Although HIV and AIDS have become amenable to control since 1997 through a multi-drug therapy that suppresses HIV multiplication in the body, HIV remains incurable to date. This method, however, cannot remove the latent HIV-1, the non-replicating virus particles that remain dormant in the cells of the body. As soon as drug administration is stopped, the viral load will again increase in the body. Therefore, a cure for HIV would mean the lack of viral activity after treatment. The kick and kill approach, which was developed several years ago, targets reservoir cells, activates the HIV virus inside, and lets the immune system do the rest. The problem with the kick and kill approach is that a satisfiable kill device has not been available yet. However, Kumamoto University's new approach, the Lock-in and Apoptosis approach, goes one step further and actively kills the targeted host cell. The proposed approach of the researchers follows the kick process and facilitates eradication of HIV-infected cells by essentially turning an HIV-infected cell into a prison cell from which the invaded virus cannot escape. The host cell will eventually die through programmed cell death or apoptosis without releasing the prisoner virus. Kumamoto University's approach utilizes the newly created synthesized compound L-HIPPO. The finding is particularly exciting because it could lead to total eradication of the HIV virus from the body, something that has never been achieved before. Associate Professor Mikako Fujita stated, "This would be a big step towards a complete recovery from HIV. We believe that our research will help to completely eradicate AIDS." The idea of using apoptosis to the treatment of diseases is not a new idea. This has even been proposed back in 1995. However, the idea of locking a disease inside a cell and synthesizing the L-HIPPO are new ideas. So how does the L-HIPPO work? PR55 gag is one of the products produced by hijacked host cells and mediates HIV-1 variant budding. Ordinarily, PR55 gag migrates from the cytoplasm to the plasma membrane and binds to a specific inositol phospholipid PIP2 in the membrane. The PIP2 and PR55 gag assembles each other in the membrane to form variants. As PR55 gag PIP2 binding triggers the variant budding, they considered that a small molecule that binds firmly to PR55 gag could antagonize the PIP2. So they synthesized an artificial phosphoinositide derivative, the L-HIPPO. The L-HIPPO was found to have a 70-fold stronger affinity to PR55 gag. To examine the localization of HIV-1 PR55 gag, it was venous fluorescent labeled inside HeLa cells and observed by fluorescent confocal microscopy. The control showed that only 25% of the PR55 cells were localized inside the cytoplasm, while 75% of the cells are in the cell membrane. But with the DL-HIPPO together with the alpha-cyclodextrin carrier, 88.1% were in the cytosol and only 11.9% in the cellular membrane. In another examination of the capability to lock, the Western blot test showed that nearly half of the HIV-1 release was suppressed for both concentrations of 25 and 50 micromoles of the DL-HIPPO complex tested. They separated the L-HIPPO into its isomers and found that L-HIPPO was more potent, demonstrating a higher binding affinity and inhibiting more than 70% of virus release. Also, the apoptotic effect of L-HIPPO was examined via flow cytometry, where the cells are stained, incubated, then analyzed by fluorescence-activated cell sorting. A dose of 50 micromoles of the complex showed that the highest ratio of the cells stained, indicating the induction of apoptosis. They have not filed any patents as of this moment, but I have found a similar patent by Chang and Goldenberg. The idea is similar to theirs. It uses a complex and at least one anti-HIV therapeutic agent. The dock and lock method is an approach that develops bioactive molecules with multivalency and multifunctionality, although the lock-in and apoptosis approach remains a novel idea of theirs. Unfortunately, their approach is not readily available yet to people with HIV. What they demonstrated in the lab is just a small step towards a cure. There is still need to improve the drugs that activate the virus. I envision a partnership with pharmaceutical companies to further develop their strategy that would hopefully advance the clinical trials. And sometime in the future, we hope to finally eradicating HIV.